Hello, and a very warm welcome to everyone to our today's webinar on comparative clinical endpoint and pharmacodynamic bioequivalence studies for generic orally inhaled drug products. I am Anvesha Bhattacharya and I represent Veeda Clinical Research. Veeda Clinical Research Limited, together with its subsidiary BioNeeds India Private Limited and its joint venture Ingenuity Biosciences Private Limited, together referred to as Veeda Group, offers a comprehensive portfolio of clinical, preclinical, and bioanalytical services to support innovative, biosimilar, and generic drug development programs for our global clientele. Together, we serve clientele globally in pharmaceutical and biopharmaceutical, agrochemical and industrial chemicals, herbal nutraceuticals, medical devices industries. So as your facilitator today, I would like to introduce you all to a couple of points as we begin this session. This session will have a presentation in the beginning, followed by a Q&A round. We have enabled our Ask a Button feature. It's right on your screen, where you can drop all your questions related to this webinar right there. Now, moving forward, I would like to introduce to all our panelists for today's session. So our first panelist, Dr. Ravi. Dr. Ravi Alamchandani is a MD in Pharmacology, gold medalist with over 6 years of experience in the field of medical affairs and safety reporting. He is currently working as a General Manager, Medical Affairs and Pharmacovigilance at Veeda. His experience includes working in various hospitals, pharmaceutical companies, and contract research organizations like DDMM Heart Institute, Torrent Pharmaceuticals, and Lambda Therapeutic Research before joining Veeda Clinical Research. Our next panelist in today's session is Dr. Hiren Mehta, currently working with Veeda as a Vice President and Head, Clinical Affairs. He is a PhD in clinical pharmacology and comes with a rich experience and deep knowledge of pharmacology and scientific affair functions with over 19 years of experience in managing clinical pharmacology functions, medical scientific affairs and writing, pharmacokinetic and pharmacodynamics, data analysis and reporting, and regulatory compliance. He worked for Alta Sciences, Syneos Health, Lambda Therapeutic Research, Torrent Pharma, Cadila Pharmaceuticals, where he was responsible for clinical pharmacology, PKPD, clinical research, medical writing, and clinical affairs from both a CRO and a pharma company perspective. Our next panelist is Dr. Ashutosh Jani. He is PhD in pharmacology, has over 17 years of experience in the field of clinical research, and is currently working as a head of clinical operation department at Veeda. He has experience working in academia, pharmaceutical companies, and contract research organizations. During his tenure, he has managed teams involved in clinical operations and business development and has good rich experience in clinical development, project management, contract negotiations, and implementation of programs in various therapeutic areas. Our next panelist in today's session is Mr. Paresh Patel, who is currently working as Head of Pharmacokinetics and Feasibility at Veeda and has 13 years of rich experience in the CRO and pharma industries. In the clinical research field for various studies for various dosage forms, such as oral dosage forms, lyophilized, transdermal patches, topical dosage forms, nasal and oral inhalation dosage forms, long-acting depot injections in healthy as well as patient population. So without any further delay, I would like to call upon Dr. Ravi to begin the presentation. Over to you, Dr. Ravi.
Yeah, hello everyone. So we'll be discussing briefly about, I'll be briefly discussing the topic oral inhaled products. So these are the drugs which are given through mouth and you expect the drug to act at lung level to treat respiratory disease. However, the therapeutic scope as of today is widening and we are treating even systemic disease through this route. Next. So before I begin into what all systemic diseases, first of all, we'll see the respiratory disease burden that our society is currently facing today. If you see worldwide, around 200 million people are living with asthma and COPD, or 70,000 subjects are living with cystic fibrosis. Around 3 million patients have idiopathic lung fibrosis, and around 10 million people suffer from tuberculosis annually. Not only this, lung cancer is a leading cause of death worldwide. Around 1.8 million deaths occur annually. Pneumonia is very common, and it is the largest infectious cause of death in children. It accounts for 15% of all the deaths of children under 5 years of age. Now, we all are aware of COVID-19, the menace it created. It was actually a respiratory disease in itself. So if I see, there is a huge burden of respiratory disease in the society, and various drugs can be developed to manage or to treat these diseases. As of today, the global respiratory market is increasing annually at a rate of 4 to 8%, 4 to 6%. Next. So what are the therapeutic areas, or what are the diseases for which the oral inhaled products are currently being used for? If we see, the most common disease for which we have oral inhaled products are asthma and COPD, for which we have beta agonists, we have muscarinic antagonists, we have glucocorticosteroids. Then there is cystic fibrosis, for which we have mucolytics, which is N-acetylcysteine. Now, this N-acetylcysteine was also widely used during COVID. Rather, all the above mentioned drugs, that is beta agonists, muscarinic antagonists, and the mucolytics, were commonly being used even for the management of COVID. And they're also being used frequently for treatment of other pneumonias as well. For cystic fibrosis, currently leukocyte DNAs are also being used to reduce inflammation, and even the inhaled antimicrobial agents like tobramycin are given to treat the local infection in the lungs. Next. So why do we need to give oral inhaled drug products? Simple. The drugs are being directly delivered to the site of disease. This results into faster action. Smaller doses are effective. The route is painless and thus helps in improving patient compliance. The systemic concentration of the drug is low, hence you have less systemic toxicities. It avoids first-pass metabolism. Thus, you can have lower doses, you can have more reliable and more predictable action of your drug. So that is the advantage of developing oral inhaled products. Next. So as you can see, when a person takes the oral inhaled drug product, around 60% of the drug reaches the lung. The rest of the drug, it enters into the GI tract. So this 60% of the drug that is reaching the lung should be adequate enough to provide your therapeutic response. And when you develop the drug, you want that minimum drug should be settled into your oropharynx and thus enter the lung, thus enter the GI tract, because this drug will ultimately result in local and systemic side effect. The higher proportion of drug reaches the GI tract, the higher chances of having the systemic side effects. Next. So the devices that are commonly being used to administer the drug through the inhaled route are metered dose inhalers, drug powder inhalers, nebulizers, and your inhalation sprays. So next. So when you intend to give the drug through the oral inhalation route, it is necessary that your device should be such that it is effective. It should take into consideration the poor patient's compliance in terms of taking the drug, because different patients are old. The method of taking drug by an older person may be different from that by an adult, and which will be different from that by a child. So your method of administering, or your device for administering the oral inhaled product, should be thoroughly characterized. Now, this characterization may be based on in vitro method, in vivo method, or ex vivo method, but the device has to ensure that there is a reproducible effect. The drug being released at each time is consistent. Next. Now, when I see the global development of the oral inhaled drug product, or the research in progress for this particular therapeutic area, this particular area suffers high attrition to the extent of 70%. That is, of the 10 drugs being evaluated, only 3 get to see the day of light. Now, the cumulative probability, among them, the cumulative probability for a molecule to reach the market is around 3%, which is very less compared to other therapeutic areas, wherein the probability of a drug reaching the market is anywhere between 6 to 14%. Now, various factors may be responsible for this, this poor attrition rate. Now, this may be attributed to drug, this can be attributed to the device, or it may be because of the poor understanding of the disease under study. So, what are some of the challenges that one encounters when one is developing these oral inhaled drug products? Let us focus on them. Next. The first challenge that a researcher encounters is the drug getting deposited in the oropharynx. Now, while the companies are trying to develop these drugs, we should ensure that there is a minimum accumulation of the drug in oropharynx. Now, this is a problem because of 3 major reasons. First, there is a wastage of drug. Then there is local toxicity. Third, this drug enters into the GI tract and can cause systemic toxicity. Next. The second thing one should ensure while developing oral inhaled product is that the drug should reach its site of action. Now, for this, 2 things are important to be considered. One is the velocity or the speed at which the drug formulation is being released from the device, and the size of the particle. Both these parameters, they impact the drug delivery to the site and the action of the drug. Next. So, both these parameters need to be adequately controlled. Now, velocity, if the drug is released at a high velocity, there is high likelihood that most of the drug will get deposited in the oropharynx or the tracheobronchial region itself. So you want to ensure that the velocity at which the drug is being released is low, so that the drug can slowly reach the distant part of the lungs, even up to the alveoli. It goes without saying that your drug cannot reach the part where, there is no flow at all. So there are certain diseases wherein there will be obstruction of certain segments of the respiratory tract and your drug may not be, reaching those sites. In such cases, you will have to combine the effective use of pharmacology wherein, the different drugs can be combined. One that can cause bronchodilation, other that can cause mucolysis, and third, the drug can reach the site. So you need to have a combination of drugs sometimes for the drug to reach its site of action and to show effect. So velocity at which the drug is being released from your device is very important while you develop the device. Next is the size of the particle. If the size of the drug particle is large, it will get deposited in the oropharynx and may not even reach beyond trachea. Smaller size particle ensures that the drugs reach, to the distant part of the lungs. It is said that if the particle size is less than 3 micrometer, then they easily reach to the alveoli by diffusion method. So smaller the size of the particle, better the chances of drug reaching its size of the site of action, and thus the better chances of it showing effect. In all, the particles, they observe the Stroke law. That is, even if the size of the particle may be large, but since these are globular in shape, their aerodynamic size is less. Next. The third important factor that you need to consider while designing or developing your drug is that every drug will be cleared from the lung. So you need to be fully aware of the elimination that takes place in the lung. This elimination may be through the mucociliary clearance or through the macrophage clearance. Now, mucociliary clearance is basically the ciliars that constantly try to clear the lungs of all the foreign particles. They ensure that all the foreign products, they are thrust towards the pharynx, and ultimately this mucus will thus then be swallowed by the person and will enter into the GI tract. So there is a mucociliary clearance. Some amount of drug, they are being broken down by the macrophages. Apart from these two major clearance systems, there is third something known as absorptive drug clearance. That is, the drug reaches into the lining of the lungs. From there, they will get absorbed into the blood circulation. Now, this practically happens from all the parts of the lung. Currently, this mechanism wherein the drug is being absorbed from the lungs into the systemic circulation is being developed or is being evaluated to treat the systemic diseases. Next. The fourth is dissolution and subsequent absorption of the drug from these particles, from the particles that have reached the lung. So the dissolution and absorption of the oral inhaled drug is highly influenced by the fluids in the pulmonary system. The diseases of the lungs, they alter the physiology of these secretions, and thereby they impact the dissolution and absorption of the drug and thus its action. The mucus layer that is there in the lung, it acts as a physical barrier for the dissolution and absorption. So in diseases like COPD or asthma, wherein there is thick mucus layer, there is impairment of dissolution and absorption of the drug that reach the lung. On the other hand, the surfactant that is present in the lung, it helps or it facilitates the drug dissolution and the absorption. So one must be fully aware of the disease being treated. If the disease is such that it increases the thickness of the mucosa, of the mucus, there will be definitely an impact on the drug dissolution, absorption, and thus its action. Next. The fifth point that one must consider or one must keep in mind is that the lung has different composition. And in all, the metabolic rate in the lung is less as compared to the GI tract. So this helps that, the drug stays in the system or drug stays in the lungs for a longer period and you get better efficacy. However, in certain diseases like COPD, there is increased amount of protease activity in the lungs. This results into the faster clearance of the disease from the lungs, and thereby lesser effect of the drug at the lung level. So while you're developing drug, you must take into consideration that if there is a chronic inflammatory condition, the protease activity in the lungs will be higher, and thereby the clearance of the drug from the lungs will be higher. Next. So we see that, when the oral inhaled drugs are being developed, there are various factors one must keep in mind. The device has to be appropriate. The release of the drug from the device has to be appropriate. The particle size has to be appropriate. And, you should take care of the clearance mechanism of the lung, the disease that you are treating, the mucosa that would be released in those diseases and all. Okay. Now, when we did a search of the trials for oral inhaled products that are being registered on CTRI or clinicaltrial.gov, we found that almost 3%, nearly 3% of the trials registered on these websites belong to the oral inhaled drug product. And we also found that these trials were not only meant for treating the respiratory diseases, but also systemic diseases. So this area, this drug development process for oral inhaled drug product is very important and should be kept in mind, and we should be thoroughly aware of the regulations and the trials that you need to do for getting these products in the market. So our next part is our next speaker, Dr. Hiren, will cover the trials that you need to take into consideration while, if you want to bring these products into the market. Thank you.
Thanks, Dr. Ravi. Am I audible, Pawan?
Yes, audible.
Okay. So good morning, good afternoon, good evening to all my colleagues and friends on this webinar today. And I thank you, Dr. Ravi, for the nice explanation of the therapeutic part of the products and how the mechanism works. Now, for any company, in order to develop the generic product of any orally inhaled product, generic or 505(b)(2) or other regulatory pathways, we just want to cover up most of the regulatory requirements for FDA and EMA. The other regulatory agency will mostly follow the same transcript that FDA and EMA has published. So today I will focus on FDA followed by EMA requirements in terms of the how we design the study, what are the endpoints, and how we want to make sure the results are communicated to the agency properly. Next, please. So as we all know, normally the bioequivalence criteria normally needs the pharmacokinetic studies for most of the drugs which are acting systemically. It's very common. Now, this drug, they normally reach to the sites of action through the systemic circulation. So basically what is there in the system is reflective of these site of action. Now, such approach may not always be considered for locally acting products, because they normally the actions are intended on the local site, and consequently they do not rely on the systemic circulation. So we cannot just have the systemic circulation approach and just conduct the case study and submit the dossier. Therefore, an alternate approach is required to be considered for most of the locally acting drugs and products for the bioequivalence criteria. Now, these orally inhaled products are a class of product, as Dr. Ravi said, most likely dry powder inhalers, DPI that we normally call, MDI, metered-dose inhaler, and some of the nebulization product that we use with the chamber. Now, most of the inhalation products are designed as a locally acting in the lungs, and therefore their drug delivery does not rely on the systemic circulation. So we have to consider an alternate approach apart from the routine pharmacokinetic studies. So in general, if we don't focus on a particular regulatory agency to provide the bioequivalence and submit the dossier, we need a couple of assessments that includes the in vitro comparison of the reference product versus the test, also the in vivo, that means the pharmacokinetic, as well as pharmacodynamics, or sometimes if it is not possible, we may need the therapeutic endpoint in the clinical study with the patients. So this is in general approach. We'll focus on the regulatory wise acceptance criteria. Next slide, please. So for FDA submission study, normally they accept the dossier using the generic route as an ANDA submission. Before we start the even product development and go to the clinical studies requirement, the foremost requirement is that the device itself, either the inhaler or the nebulizer chamber or the metered dose, should have a similar characteristic in terms of device as well as the release rate from the device itself compared to the US market reference product, which is, I will just call them as URP in my next speech. Apart from the device similarity in terms of the performance of the device, the organics of the device, the mechanism of the device, it has to be also similar in vitro characteristic compared to the URP. After we prove the device similarity as well as the in vitro performance between the test and the URP, for a similar safety or similar exposure, we have to prove that the systemic exposure, even though using the alternate route through the lungs, the systemic exposure should have to be matched with the reference product itself. And after that is done, the other approach that needs consideration for the FDA is the similar efficacy in terms of the therapeutic equivalence. Either we can use a pharmacodynamic biomarker or a clinical endpoint in the patient study versus the URP. And most of the case, we'll end up using the placebo for the study sensitivity as well, and sometimes using the parameters for the placebo correction. Next. So this is a typical approach for the FDA ANDA filing for generic product of the orally inhaled products. So they basically focus on 4 different criteria. So the first one is, as I discussed previous slide, formulation and device similarity, followed by the in vitro equivalence in terms of the dissolution and other parameters for these kind of products. Systemic exposure, of course, the PK endpoints using the healthy subject study, followed by pharmacodynamic or clinical endpoint efficacy equivalence. So for a CRO, like Veeda or somebody else who is mostly doing the healthy BAB studies, we will focus on the last 2 options, which is systemic exposure plus the PD or clinical endpoint efficacy studies. The first 2 normally is being taken care by the formulation team, and there are some expertise in the market which help you with the in vitro equivalence as well. Once we complete all these 4 criteria, then only FDA normally accepts the dossier. Next. So the typical in vivo PK bioequivalence study design will be normally a fasting condition, single dose and 2-way crossover, and comparison of the systemic exposure between the 2 drug products. In terms of selecting the strength, when we have multiple strength in the development, normally all the strengths should be tested because the relationship between the PK dose proportionality across all the strengths is not yet known. Only the in vitro performance parameter are known, but that normally doesn't translate into the dose proportionality using this orally inhaled route. So most of the time we end up using all the strengths unless there is a strong rationale why we should only go with the single strength. The selection of dose normally should be sufficient number of inhalation that is enough for the characterization of the levels into the systemic circulation using a sensitive analytical method. Normally, we select the adult males and non-pregnant females in a very generalized population. The acceptance criteria for the PK endpoint will be normally the Cmax and AUC. The 90% confidence interval of the geometric mean ratio should fall within the 80 to 125%, unless there is an widening criteria applicable for the drug product. Next, please. And these are the detailed description of the endpoints for this kind of, once we clear the PK endpoint study, then we jump on to the pharmacodynamic or clinical endpoint study. These are the typical study design description criteria for PD or therapeutic equivalence study. Normally, it is randomized, as I said, placebo control for the study sensitivity, and sometimes placebo-corrected values also needed. We can do it either parallel or crossover. We can call it comparative clinical endpoint or PD endpoint study. Normally, we have a placebo run-in period for all the treatments, followed by the treatment using placebo, test, and reference product. As I said, the study sense for checking the study sensitivity, we'll need placebo treatment, and also some of the parameters may need baseline or placebo-corrected response. The objective of this kind of study will be mainly to determine the difference in the local delivery at the site of action between the drug products, because the PK normally doesn't translate the PD response considering the route of administration. In terms of selection of strength, when there are multiple strengths to be developed, we normally prefer the lowest label dose with the lowest strength, unless there is a specific regulatory requirement. In terms of number of doses to be administered, normally single dose are sufficient, but sometimes we may go up with multiple doses based on the mechanism of action, and also sometimes depending upon the patient population. The selection of study population mainly goes in a direction of the indicated patient population in the approved labeling. Any one of the indicated patient population is sufficient. For the bioequivalence criteria for this kind of clinical endpoint or PD endpoint study, normally we use the dose scale analysis. And for the clinical and PD endpoint, normally the endpoint, the geometric mean ratio for the test and reference should fall within the standard limits of 80 to 125. Or if it is purely a PD biomarker study with the effect curve, based on the area under the effect curve AUEC, the 90% CI confidence interval for the relative bio should fall within 67 to 150. So this relative bio is compared to the reference product using the dose scale analysis for the PD biomarker. So we have to prove two criteria. First one is for the clinical or PD endpoint, and second one using the dose scale analysis, the relative bio of test versus reference should fall within 67 to 150. Next, please. So that was for FDA. EMA has a slightly different approach for the orally inhaled generic development, and they normally don't accept the simple generic application. They normally call it hybrid submission. Of course, there should be a device similarity, just like the FDA requirement between the test and the European marketed product. In vitro performance, of course, should be matching with the characteristic of the reference product for the test itself. And for similar safety or exposure study, they recommend two kinds of studies. One is without the charcoal block, and the other one is with the charcoal block. So when I say without the charcoal block, normally the objective will be the test product must follow the similar systemic exposure in a PK study versus the reference product. So when we don't have a charcoal block that is similar to the FDA PK study, we measure all of the systemic exposure either through the lungs, through the local site, or by another route. And for efficacy, they suggest using an activated charcoal block. So normally the use of activated charcoal is to block the systemic absorption. So whatever you see, either in terms of PD marker or the PK, it mostly through the local site. And that's why they recommend using the charcoal block. So whatever we see is only based on the site of action. Next, please. A little complicated flowchart here, but try to make it simple. The first and the foremost requirement for the EMA to generate a generic of the orally inhaled product, first of all, they should be in vitro equivalent in terms of all the characteristics required for the device. And then pharmaceutically equivalent, that means all the excipients, the ratio, all the other characteristic of the product should be equivalent. In that case, we can simply just demonstrate the bioequivalence if it is both in vitro plus pharmaceutically equivalent. But most of the cases, it is difficult to prove the pharmaceutical equivalent due to unknown characteristic of the excipients and the ratio. So that time we normally go for systemic exposure study, which is a PK endpoint study, just like an FDA. Now, if we have a positive in vitro equivalency, but we can't prove the pharmaceutical equivalent, and there is a less possibility of the PK study, we can also conduct the lung disposition study. I think either using the image part or the just replace that with the simple PK study to check the deposition of the drug using the device in the lungs compared to test and reference. Once we prove the PK study that the test and reference are matching in terms of the systemic exposure using both charcoal block and without the charcoal block, We have to move to the next level of the PD endpoint study, where we have to prove the clinical endpoint in the patients. So similar to FDA, they also most of the time need PK plus PD study, unless we are able to prove an alternate approach using the lung deposition study, or we are able to prove that the product is pharmaceutically equivalent plus in vitro plus PK equivalent. Sometimes we are able to skip the PD endpoint study if the other criteria are matched with the reference product. Next slide, please. These are the typical EMA recommended study design for the PK plus PD endpoint studies. The first few rows are for the PK endpoint study. So normally, similar to the FDA, we go for single dose 2-way crossover PK endpoint study with two separate arms of either charcoal block and without charcoal block. So we can have a 2 separate study, or we can conduct a 4-period study. 2 of the period they will have charcoal block, 2 of the period they won't have a charcoal block. So we'll compare the total systemic exposure, and we'll also compare the systemic exposure with the charcoal block, that is only through the site of action, site of application. The objective of this kind of study is normally to check the equivalency in systemic exposure between the drug products with and without charcoal block. Normally, EMA only asks for the single lowest strength if we are able to investigate the dose linearity using in vitro method for both test and reference. If the reference is not dose linear but test is dose linear, then also we need other strength. If both are dose linear, then we can just have one single strength, the lowest one. If there is no information on the dose linearity for either test or reference, bioequivalence criteria should be proved with one more strength, possibly with, if possible, all the strengths, otherwise at least the lowest one and one more strength, if there is no information on the linearity of dosage form. Of course, the dose selection based on the minimum number of inhalations required sufficient, which are sufficient to characterize the plasma levels using the sensitive analytical method. Normally, we suggest adult male and non-pregnant females, a general population for BE study. And again, the bioequivalence criteria for PK endpoint study normally is the 90% CI for Cmax and AUC within 80 to 125. EMA sometimes asks for partial AUCs as well, based on the product characteristic or the device drug release pattern. So that is about the PK endpoint study for EMA. For PD endpoint study, EMA has not published, to the best of my knowledge, any specific guidance, but normally we have seen that if we follow the FDA recommendation for EMA, that should suffice in terms of study design and the endpoints and the assessment using the dose scale analysis. Next slide, please. Okay, so I think that's what pretty much it for my slides in terms of the regulatory and study design requirements. I will hand over that to Dr. Jani for the further slides. Thank you.
Yeah, thank you, Dr. Hiren and Dr. Ravi. So I think as Dr. Ravi rightly explained, the physiology part and what the drug would be doing to the body, and Dr. Hiren more onto the regulatory front. But to do this, definitely there are challenges when it comes on an operational level. So what we'll be doing is we'll be just going through the operational challenges associated with the conduct of these type of studies, and what could be a proactive mitigation plan so as to we minimize all the challenges associated with these type of oral inhalational studies. Yeah, we can go to the next one. Yeah, so complexity and challenges for the locally acting oral inhalational drug products. So the first of them is the dose selection, the subject selection. Like say, dose selection would be based on the systemic concentration because it would be very low, and we also would need a standard method for its detection. Similarly, we'll also need to select whether the study need to be conducted in healthy or it could be in a diseased patient. The physiology would be totally different when it comes to the subject selection. The patient training, the device related issues or the designing with respect to the same for the easy giving of the inhalational product, and the formulation because the properties, the physiochemical properties and the ingredients would all be affecting the total availability of the drug onto the systemic circulation. Yeah, we can go to the next slide, please. The next one. Yeah, so for all these, definitely what we need is an experienced team. So we at Veeda have an experienced project team, and the project manager is usually the one point of contact for these type of studies, and there's a strong management oversight from the senior management, and the project manager is accountable for the project delivery. That includes all the reporting, the status, all the timelines associated with the study, including managing of the study finances. The support functions like report writing, data management, biostats, he gets all the work done through them and delivers to the sponsor in a timely manner. The project manager is supported by colleagues of him like the CTAs, CRAs, the regulatory expert, and the medical monitor team. We can go to the next one. So as you've already seen, and these type of oral inhalational product studies, they come up with a large sample size. So conducting them in patients, it would be a long study duration. However, we try to cut short the duration as much as possible through our accelerated startup, where we try to engage early so that the protocol consultation, the facilities associated with the project, the resource planning, all these activities are taken up very early on the drug development stage. And we leverage our expertise and rapport with various ethics committees across hospital sites in India. And through this, there would be a rapid approval from the independent ethics committees. And we already have specific templates which could be helping in planning of all the other activities associated post approval of the study. So we have a system of a clinical trial management system through which we could fast track the recruitment and get a clear overview about the possible recruitment across various sites. And we've been successfully able to get the first site approvals within 6 weeks or less. So that's what our standard is. So as soon as we are awarded the study, we try to minimize the time with the accelerated startup system at Veeda. Go to the next slide, please. The key success factors, especially when we considered the studies, it's important to understand the nuances of the respiratory trial. And as Dr. Ravi and Dr. Hiren lightly explained you the physiology and the regulatory requirements, a knowledge and experience similar one for the proposed site is equally important. We have highly experienced and effective project management team to take care of these type of studies. Where there is a necessary of close collaboration with the site to have the effective recruitment. As I mentioned, the sample size for these type of studies is large, and so we be enrolling a very large amount of patients across various sites. Sorry, can we go to? Yeah, thanks. So the other important aspects would be more onto the training, the data handling, and planning with respect to the site enrollment and the retention, which is equally important as far as these type of studies are concerned. Yeah, we can go to the next slide. Yeah, so when we consider the challenges, so we just try to divide the different challenges associated with uh, oral inhalational product studies. Is there could be some site specific issues or challenges. There could be study specific, patient specific, or based on the current medical practice of the investigator. Now, in site-specific challenges, what we have is the lack of awareness about spirometry. Now, the investigators are routinely doing spirometry just for their clinical practice, but when it comes to clinical trials, it's a different ball game altogether. So their awareness with respect to this is very essential. Uh, the sites would be lacking calibrated or validated documents with respect to the spirometry. So they may be having spirometer, but the equipment would not be calibrated or validated. The quality of spirometry graph which are produced through the meters, they should be meet the American, the European standards for spirometry. And as I mentioned, there are large patient enrollment which are supposed to happen across sites. So the site should be equipped with handling a large number of patients during the course of the study. There are some study-specific challenges as well. So based on the different type of study, there is the need for a rescue medication, because these type of studies come up with a run-in period where you'll try to remove the previous effects of whatever the medications they are on. So getting the patient off the medication, getting them on a run-in period, and providing them certain medication based on the rescue, that is an important part with respect to these type of studies. And the site staff need to stay for a longer time, because when it's clinical trials, there are different time where we would like to have the spirometry or the PF1 calculation to be done. So for these type of studies, we anticipate that there could be long stays for the site staff. The validation is also important. So because what happens is these type of inhalers, maybe a powder dose inhaler or meter dose inhaler, would be used by self-administration. So the patient should be well trained onto using of these devices for the exact result to be obtained. So as I also mentioned, there is extensive run-in periods, and these are the periods when we wean off the patients from their active treatment, what they are undergoing, and these periods are really challenging periods. The other thing, specifically with respect to study, are repeat serial spirometry. So in the normal practice, they may not be doing, but especially when it comes to clinical trials, it's necessary that we get the best spirometer or the best spirogram for the evaluation of PFE1. So there could be repeat of these which needs to be done, and that could be home-based monitoring, which could be also required. Certain challenges are is patient specific. So there are various factors like environmental factors, the smoking history, the genetic factors, which play a vital role in the patient selection, because the amount of cigarettes which they are smoking, their history with respect to smoking, etc., plays a vital role, especially when we come to the outcome of the study. Especially in India, where there are different forms of smoking available, like say BDs, etc. So calculating the total size is also equally important. Education of the patient is really vital, because they need to take the drug in the right way. And the regulatory requirements, as we already seen in the previous slides by Dr. Hiren, they would also be slightly different when it comes to the patient studies, maybe US and Europe. The symptoms diary, the exacerbation happening during the course of the study, these all needs to be captured in the diaries. So the patient need to be trained to calculate and document these exacerbations during the course of the study. The certain challenges with respect to the medical practice are also there. So the patient medical records are not usually up to date. So at certain places, there no practice of maintaining the medical records. And the past medical management would be different based on the site to site variation would be there. And there is not a good practice to write detailed notes. So there'll be just short notes which would be written by the investigators, and it's at times very difficult to interpret what exactly treatment or what the condition of the patient is. And as I mentioned, the BD smokers are also more when compared to any other country. So calculating the exact amount of the consumption of BDs or converting the BDs into cigarettes, we need to ensure that that's correctly formulated. We can go to the next slide. So what the proactive approach we at Veeda follow is we lay very good emphasis onto the training. So there is an intensive training of these investigators and the study site personnels which happens during the initiation visit. And we ensure the proper logistics and the robust samples are being shipped to the respective sites. The site infrastructure is also of utmost importance, especially having uniform equipments across all the site, because the spirometry and the spirogram varies from make to make. So it would definitely be advisable that we have a uniform make of spirometer across all the sites. So for this, we may have to rely on to providing a uniform spirometer to all the sites. The equipment needs also be validated and calibrated at the site. So multi-vendor validation and qualification should be ensured to get the proper IQ, QPQ of this done as per the protocols of spirometer. And the sites should be equipped to upload these spirograms from this parameter directly onto the central depot. The other challenges are with respect to shipments. The trial runs from site to site are done, so we get a fair idea about what could be the transit time for the shipment to reach the site, and the site MPs are reaching on the right time. As I mentioned, calibration, we also provide the calibrated spirometers or maybe calibrated clocks to get the exact timing with respect to the spirograms and the conduct of the study. Yeah, we can go to the next slide. So as we've seen in the previous slides, that the technique or the inhalation may be dry powdered inhaler or meter dose inhaler. The technique is of utmost importance, because this would be determining the total outcome of the study. If it's not been properly done, the outcome of the study would be varying. So what we do is there's a check of the dose counter is there. So if required, there should be you need to shake the inhaler if it's instructed under the protocol, and the priming the device should be equally be seen that we prime the device ready for use. The next step would be the exhalation. So the patient should be trained or should be trained for exhalation gently from the mouth, because there is this exhalation which happens slowly from the mouth, and the mouthpiece is put up on the device mouthpiece is put up on the mouth, and the tin is slightly tilted and closed the lips. So the patient is instructed to close the lip tightly around the mouthpiece, and then there would be inhalation which happens. So depending upon the type of inhaler which we are using, maybe dry powder or meter dose inhaler, this, if it's a dry powder, it should be quick and deep, and when it's a meter dose inhaler, it should be slow and steady. And subsequent to the inhalation, you need to hold the breath. So after holding the breath, you have to remove the inhaler from the mouth and hold the breath up to 5 seconds. So these would be ensuring that all the inhaled drug particles are reaching into your lungs, and the absorption happens through this route. And then the closing the inhaler cap and repeat if necessary. So these activities, the patients are thoroughly trained onto this inhalation technique, because this is the one which plays a vital role and would be affecting the study outcome altogether. We can go to the next slide, please. So the operational risk and proactive mitigation plans have been put up. So we ensure that the exact date of birth needs to be calculated, is essential to calculate the predictive values of the spirometry. So we rely on to getting the age proof and the documents during the screening itself, so that we get a proper age proof for these patients. Consistent interpretation of the spirometry reading by the reviewer is important. So in case of there is poor quality which does not meet the required requirements, the patient may be enrolled, but ultimately would be ineligible for evaluation. So this interpretation of spirometry is equally important. Non-cooperative and efforts from the patients and technicians are not performing the test correctly. So what happens is, like say, we fail to meet the requirement if the patient does not support, or the technician is doing a faulty exercise. The total patient may not be usable for the study. Use of incorrect race or the correction factor. So as I was just mentioning, there's more prevalence of PDs in this region, and that needs to be converted into the cigarette pack years. So that should be a correct formula which will be used, and that's equally important. So what we do is, like say, we use the age proof document itself during the screening. There is an effective oversight for the monitoring team. So where we request the central spirometry vendor to send a copy to the monitor, so which has been perfectly validated and correct. The spirometry will be performed as per the American Thoracic Society and the European Respiratory Society standards, so that the patient is not excluded from the study, and the requirements have been fulfilled. So there is no need to identify there is a need to identify sites that will have this well-trained investigators and technicians. So with these, we shall be able to take care of the study requirement. The training and qualification of these technicians by the central reader organization would definitely help in reducing the variability, especially with this and the screen failures during the course of the study. We can go to the next slide. Amisha, we can do the next slide, please. Thank you. So as I mentioned, at Veeda, we have these capabilities, and Veeda could act as a one-stop shop for all your study-related requirements. I think that's it from my end. Thank you. Thank you, everyone.
Thank you. Thank you so much. Thank you to all panelists for this wonderful presentation. Now moving on, we will be proceeding ahead with our discussion, where we are going to answer some of the interesting questions posed by our audience. So, Mr. Paresh, my first question to you is, for FDA submission, if the in vitro profile comparison does not meet the matching criteria, can we go ahead with the PK and clinical endpoint studies? And if passed, can we submit with a rationale?
If in vitro is failed and PK plus PD are meeting the bioequivalence criteria, even though we cannot move ahead for FDA submission, because all three studies should be meeting the bioequivalence criteria, then only we can conclude that our test product is bioequivalent with the reference product for oral inhaled dosage. So it is not. If we try to submit, then possibility that OGD refuse to receive our dossier.
Okay. My next question to you is placebo treatment mandatory for the PD endpoint studies?
Yeah, because PD study itself is a high, insensitive method to detect for bioequivalence. So to reduce the baseline variability, regulatory recommend to have a placebo arm in the study for clinical as well as PD endpoints.
Okay. My next question is, when will we need the multiple dose study? Is it required to demonstrate the steady state at systemic or local level?
If it is a PK endpoint study, then we require to demonstrate a steady state for the local and systemic availability of drug. But if it is a clinical or PD endpoint study, we are doing after multiple dose administration, then in that scenario, we don't require to measure any steady state reach or not, because our endpoint are clinical based on our efficacy response.
Okay. So my last question to you is there any alternative to charcoal block study? Can we use a different agent to block the systemic absorption?
So there is a, as per EMA guidance, if we want to waive the charcoal block study, there is a one option is that generally this charcoal block study is recommended to distinguish the systemic viability of drug by GI route and locally drug availability at the lung site. So if drug having a early deposition in lungs, like in around 5 to 10 minutes, drug are available at lungs only, and then after when we are getting any systemic viability is based on that GI tract. So in that scenario, EMA have a one option that we should add one partial UC parameter like 0 to 30 minutes in our PK endpoint study without charcoal block study, where we can show that our early AUC, what we are getting during first 30 minutes of after inhalation, is only based on lungs viability rather than any involvement of GI route absorption. So by proving partial AUC in additionally other parameters, we can waive the study after conducting only without charcoal.
All right. So thank you, Mr. Paresh, for answering almost every questions which were posed by audience. I would like to thank all the panelists for nicely answering most of the questions and taking a pause over here. We understand this session has kept us well engaged throughout, but due to some time constraints, we are not able to take any further questions at this point of time. But please be rest assured that we will be sending answers to every questions yours via email. Also, we will be sending across the recording link of webinar on all registered email IDs. Lastly, on behalf of Veeda Clinical Research, I would like to sincerely thank you all for being here today and taking out time to be a part of this webinar. I would also like to extend our gratitude to all the panelists for being such a wonderful presenters and giving us all great learnings today. We at Veeda Clinical Research earnestly look forward to hosting you all. Till then, take care and stay safe. Thank you. Thank you so much. Thank you, Dr. Hiren. Thank you, Dr. Ravi. Thank you, Dr. Ashutosh.
Thank you. Bye.
Thank you.
Thank you.
Thank you.
Thank you.